small sequence buried in these huge, large repeats that might look like that small sequence in budding yeast. That might be a critical sequence. Or it could be that it's these larger repeats themselves that specify where kinetochore binds. So think about these two possibilities for a couple of minutes, or come up with your own possibilities. And let's talk about what you guys think and how you might figure this out. So take a couple seconds, take a couple minutes.
I can record the lectures, and then when I watch them, it'll just be a cat talking to me, which is like, or it can be this guy.
You just not only think about what the possibilities are, but how you would figure it out.
What is this?
How would you figure it out?
No, it like takes the audio and then makes the baby talk.
Can you hear us now?
Yeah, because like anytime, that means it's picking up sound. So here, wait, stop. Wait, just don't talk.
What the?
So, anytime he talks, dude, this is gonna be a good way to study. I had kids in high school who looked like that. I had kids in my high school.
Do you like them? Were you friendly with them?
Yeah.
Do the alien?
Oh my god, that's really scary.
Look, wait. Looks like Akshay.
It does look like Akshay. Let's add. That's disgusting. That's disgusting. Morning hair. I didn't even comb my hair today.
Your face.
I can't switch it back. Switch it back. I want the baby. Yes.
You know the answers?
What?
For this question.
How would you go about this? Well, you'd sequence them.
I would actually die.
And then maybe you could probably just see what proteins and stuff are in it.
What's kinetochore?
You could use like cDNA to see stuff.
What do you get on the test?
114, I think.
114.
Yeah. Dude, he got a 70%, right?
I got 65.
So one way to figure out what the structure of the kinetochore is, how would you do that? So you're saying that you could crystallize an interesting idea, difficult for a number of reasons, but an interesting idea. One thing, yeah, figure out what's the structure of this thing, and why structure? What's that going to tell you? Structure function. I like that. Yeah, I like the first part. I'm struggling with the second part, so we'll just forget about that one. All right. So the first part is start cutting off sequences. This is exactly what people have been doing. Now, it's really hard to do in a mammalian cell or in a human cell to actually go in and cut off these sequences and then see if they're functional. You could assay function by whether or not they bind a kinetochore, or you could assay function by how stable the chromosome was. But I like that idea. So how would that distinguish the two possibilities that we talked about. One is that there's some specific sequence buried in this huge array of repeats, or that it's the repeats themselves that are important. Anybody have an idea?
What? Devin told.
Yeah, that's great. That's exactly right. People have been doing exactly that. So you guys are already ready to work in a research group. Anybody else
Wait.
have an idea?
What?
Can you repeat? I couldn't quite hear what she said.
Yeah, what she said is, if you start knocking out little chunks throughout that thing, and you hit a chunk that makes that thing inoperable, whether it doesn't bind a kinetochore or whether it doesn't segregate well, you've probably found a specific sequence. If you start chewing off those repeats and any one of them kills the activity, then those repeats are necessary. All right, great. I mean, it's exactly right. Anybody else? I'm missing one thing that you guys have the skills to think of. Yeah. That's exactly what I was looking for. So, yeah, you can say that.
Does she realize that doesn't work?
Right, so one possibility is if you could sequence,
She was part of that.
and we have the entire human genome sequence, right, Greg? Right? So if you could get good sequence going through this 100 kb of repeats, and maybe you found some sequence that wasn't repeat, and you could compare that to the sequences of other chromosomes even, or across different species, you could look for some kind of homology. You look for some region that looks the same, whether you're in a mouse chromosome or across all the human chromosomes. And that might tip you off that this sequence is some very, very highly conserved core element. Yes? Yeah, so the question is, great question. So if the centromeric sequences are different on each chromosome, these repeats, are the kinetochores different also? Not as far as anybody can tell. Same sets of proteins. All right, so there is, I know you guys hate this, there is no right answer here. The answer is we don't know how this works. There are a couple of different groups here at Duke who are working on this problem.
It seems inefficient.
Contemporary research
Not gonna all
problem.
work together.
One of the problems with the sequencing approach is it's very, very hard to sequence through these repeated regions. You can't, it's very difficult as you go through repeat after repeat after repeat to assemble them and know that you didn't miss some 100 base pair sequence stuck in the middle. The other assays that have been done, it's like I said, it's really hard to actually take chunks out of a centromeric region and put it back into a cell. But nature actually does this for us. There are certain cell lines that have natural breaks right within the centromere, and then they can look at those centromeres and see how functional they are and what's left over. So what we do know is we appear to be able to take chunks of those repeats and get rid of them, and they still work. And that doesn't mean the repeats aren't important. It just means that maybe you only need some of those repeats and not all of them. Okay? Yeah? So the very smallest repeats are in the kind of 180 to 220 base pair range. And then those assemble into larger repeats that are then themselves repeated. So it's called alpha satellite DNA. So, and all of those arrays, they kind of, these repeats, these small repeats, hook together into larger repeats, and then you have these huge arrays of them. So in the end, we still don't know the answer. The two possibilities are there's some specific sequence we haven't found yet that maybe needs these repeats on, hanging off of it. Or, as somebody said, and I love that answer, people have been thinking about this for years, it may actually be that these repeats fold into some kind of wacky structure, and then the kinetic order is recognizing the structure of these repeats and not the specific sequence. It's a great idea. Yeah. Yeah, it's really hard to look at the structure in a cell that you haven't manipulated in some way that might screw up that structure. It's very difficult to look at the structure of large stretches of DNA. And they do have an odd structure, but we don't know that that structure has been specified by those repeat sequences. All right, enough of the centromeres. All right, so telomeres protect the ends of linear chromosomes. I'll say this again, DNA ends are highly unstable. Cells do not like to have free DNA ends in the cell. They'll either start chewing them back or they'll stick them somewhere. They can do this by recombination, or if you have two ends, they like to stick them together. Think back to that chromosome evolution slide we had for all the primates. There were a couple of cases where chromosomes fused. This is not uncommon. Now, it's not every day. It's not uncommon on an evolutionary scale, I should say. But chromosomes can fuse at their ends. This happens. And this can happen if telomeres are damaged. All right, so exonuclease is recombination. Ends get jammed in, can get stuck into the middle of a chromosome. There are enzymes that do this, and Greg will talk about this during DNA repair. When a cell sees free DNA ends, it thinks, I've broken a chromosome, let's fix it. All right, so what do telomeres do? Telomeres, again, are repeated sequences. They're not junk DNA, but they are functional. These telomeres, you find these long stretches of repeats. And this is the repeat for, I'm guessing, I don't know, probably human. Probably human, and they vary in different organisms. Okay, so not the same sequence all the time. And they're found at the ends of chromosomes, linear chromosomes. So how do they protect the end? They protect the end by folding into some wacky structures. And basically what they do is they protect these ends by folding into a loop. And there's kind of two loops that you can see in telomeres. There's a T loop here and a D loop here. All right, now notice this end is all kind of protected in that loop. So they form this wacky structure. And these loops structures are stabilized by, what might you guess is stabilizing these loop structures? Anybody?
Proteins.
Yeah, you got it. These telomere sequences, these telomere repeats bind a whole bunch of different protein complexes
Too much coffee.
that latch onto these things and wrap up this end in a structure and protect those ends so the cells don't see a free
Chub.
DNA
I
end.
have Tourette's.
All right, so there are proteins that are specific to the telomeres. They bind to these telomeres, wrap up that end so it doesn't get chewed back, or so it doesn't end up fusing to some other chromosome. Yes? This is the end of the DNA. So the telomere are the sequences found at the end of the chromosome. So we have all of these different repeated sequences over and over and over. This is the end of the chromosome. So in this picture, and there's an overhang here, right? So there's some single-stranded DNA right here. That's what the end of the chromosome looks like. And then that D loop and T loop are all wrapped up in protein complexes that protect those ends. Questions about the structure or the function of the telomere? Yes. So there tends to be a 3' overhang, and that has to do with how telomeres are themselves maintained. And we'll talk about that, I think, in a little bit here. Yes, okay, let's talk about that now. So telomeres, aside from protecting the ends of chromosomes, are well known to be involved in cellular aging. Telomere sequences themselves are not all replicated by the normal machinery that replicates DNA. There's a special enzyme called telomerase, which adds telomere sequences onto the ends of chromosomes. All right? So this enzyme is actually increasing the length of chromosomes by adding these repeated sequences onto the ends. If you express telomerase, and I cut a double-stranded chromosome right in the center, and I generate a couple of free ends, telomerase will start adding telomeres to those ends. Its function is to extend the chromosome with these repeat sequences. Questions about that? It's very important. You don't understand. You're not. What are you not understanding? We'll talk about that in a second. That's the mechanism. We'll talk about what aging means in a second. So telomerase, yeah. Yeah, so telomerase can add telomere sequences to an existing telomere or any double-strand DNA. Because it protects the end. Okay? Want these caps on the end to protect the chromosome. There are organisms that take their chromosomes in some point during development and start chopping them up into little pieces. These organisms are full of telomerase. This is how they discovered them. Oxytrichia is one of them. And they add telomeres to the ends. Yeah. We're going to get to this. All right. Telomerase. So, somatic cells, most of the cells in your body, do not express telomerase. All right, so most of your cells are not expressing telomerase. You'll find them in stem cells, you'll find them in the germline. But most of the somatic cells in your body do not express telomerase. So what that means is, and we're going to get to why this is when we talk about replication. It's too complicated to explain now. But DNA polymerase cannot replicate all the way out to the end of your chromosome. This is one of the reasons you have telomerase. What that means is every time your cell replicates and divides, those chromosomes shorten. And they're shortening the ends. So the telomere sequences are getting shorter, shorter, shorter if you're not expressing telomerase. As these telomere sequences get shorter and shorter, they get to some critical length and the cell will undergo what's called senescence. It's basically death. It can no longer divide. So it's the length of these telomere sequences. When they get too short, the cells arrest. They can no longer divide. So it's these telomere sequences that define the lifespan. So most of the somatic cells in your body can only go through, I think it's about 10 to 15 divisions once they stop expressing telomerase before they're going to arrest. Okay? Yes? Is there a reason they stopped expressing telomerase? There is. They shut down the machinery that is normally involved in telomerase expression. And this brings up a great question. Why do we do that? If that limits the lifespan of cells, why do cells stop expressing telomerase? Now, 10, 20 years ago, when people started to understand telomerase, a number of companies started popping up in the Bay Area. And one of their interests was in telomerase in terms of its ability to extend lifespan. So what if we could turn it back on and extend the lifespan of the cell? Would that then increase the lifespan of the organism? So I want you to think about this. Like I said, companies were built to address this. Is this a good idea? Take 2 minutes and talk about it, and then we'll come back and hear what you guys think.
No, that's how cancer happens. It'll lead to
Why? Explain.
well, I can't really explain it because I don't really know enough about it.
But I think, telomerase kills good cells, but the lack of telomerase kills good cells, but it also kills bad cells. And a lot of cancer cells just have really long telomeres and no telomerase, so that it allows them to divide forever and overspread into other cells' area. Maybe. I don't know if that's true. I mean, he'll give a better explanation, but I know the answer is cancer. And no, it's not a good idea because cancer is bad. Yeah, and it makes tumor cells. Ugh, I could have just read the next slide.
Tumor cells are immortal.
Wait, so he said here that somatic cells don't express the telomerase genes, but then he said, wait, so how do they get telomerase?
It's in the stem cells.
So,
Discussing. We're going to call you.
I really don't like this stuff.
It's found in fetal tissues and germ cells.
Wait, but he said somatic cells can only go through 10 to 15 divisions when they don't have telomerase. Does that mean every cell in our body only goes through 10 to 15 divisions?
Why are you asking?
Wait, I'll just ask the TA. Excuse me, can I ask you something? So he said that somatic cells don't express telomerase, but then he said they can only go through 10 to 15 divisions before, I forget what I was going to say. Wait, I forget my question. Never mind. That's so embarrassing.
You hold that closer, I can't hear you.
Let a cell replicate, then there is increased chance of the mutation.
Okay.
That would probably
No, I think that's a legitimate idea, right? So, I mean, we'll talk the last 2 lectures in this course. We're going to talk about cancer biology and cell division. And certainly cancer is a disease of the aging, and part of it is that it's about accumulating DNA damage over time. All right? So I like that answer.
Oh.
Right, so if we extend our lifespan, so there's, I mean, one idea, extending lifespan, you know, if we all live to age 500, is that a good idea? Are we adaptable? Yeah, there's all kinds of great kinds of ecological thoughts about, you know, what happens when you extend lifespan to that degree. Anybody else? Is this a good idea? Yeah, right here. And then up in the back. Allison, can you get the guy in the back? I'll just repeat her.
Where's the guy in the back?
Yeah, that's actually a great answer. So there's something called programmed cell death, also known as apoptosis. We're not going to talk about it in this course, but during development, it's important for some of your cells to die. It works by a different mechanism than aging, but still, you know, that's a potential issue. Yeah, Tom. Can somebody yell that out? Because that microphone's worthless.
Wait, that didn't.
If you increase the lifespan of a cell, that means it can replicate more and more. So that would help if you needed something for the medical reason, like for example, you use cells that keep replicating to help us get
Right. So this kind of gets to the whole argument for stem cell research. So stem cells express telomerase. They're immortal. So being able to have stem cells to do things like regeneration or for some medical reasons. Yeah, great. Love it. So here's my question for you guys. If I extend the lifespan of a cell in my body or cells in my body, Does my organism live longer? Do we die because our cells all senesce? So could we increase the lifespan of an organism by increasing the lifespan of the cells? There's a number of things that play a role in the lifespan of an organism. You guys have probably heard about caloric restriction. You can increase the lifespan of mice by basically just borderline starving them. A lot of organisms work this way. So there are a number of things that play into the lifespan of an organism. This may or may not be one of them. I can think of another reason why this is a really, really bad idea. One of the issues with tumor cells is they're immortal. They're immortal because they start expressing telomerase. So imagine there's a reason that tumor cells divide and divide and divide and divide and make these tumors because they never senesce. So one of the things that tumor cells have to acquire before they can be a tumor cell is they have to become immortal. They do this by turning on telomerase. Yeah, Tom.
So if you figured out how to turn it on, can you figure out how to turn it off?
That's an awesome question. Now you guys are thinking like biologists, right? So if tumor cells, if part of what a tumor cell does to be a tumor cell is be immortal and turn on telomerase, wouldn't that be a great target for anti-tumor therapies? Yeah, absolutely. And there are certainly people who are working on that. So if we start turning on telomerase in our cells to try and extend our lifespan, we're part way to making tumors. I don't think this is a great idea. Yeah, real quick. So the question is, what's the difference between a tumor cell and a stem cell? Where there are a bunch of different other characteristics. To be a tumor cell, it's not just about being immortal. It's one of the things that you have to acquire to be a good tumor cell. We'll talk about all the different phenotypes that you need in order to be a good tumor cell, which of course for us is bad. Other questions? Yeah, quick. How does it get turned off? At what point? During development. It gets turned off during development, right, in the progression from being a stem cell to being some tissue. We're not going to talk much about development. Bio 119 is your ticket. That's where you learn about that stuff. Other questions before we leave telomerase? We'll come back to this too in the last couple of lectures when we talk about cancer biology. All right.
I hate CR so much.
So we're going to switch gears. We just got done talking about functional elements on chromosomes that are not junk DNA and are not genes. Now we're going to talk about chromatin. And chromatin is the DNA plus proteins. And we're going to talk about the functions of chromatin. One function of these proteins that are stuck on the DNA is to package or condense that DNA strand so it fits in the nucleus of the cell. Without it, these strands are huge, and they would never fit in the nucleus. So if we start with prokaryotes, historically, prokaryotes are not thought to have chromatin. They don't have the same kind of chromatin as eukaryotes do, although they do have proteins that stick to them. And again, this is an E. coli chromosome. See supercoiling stuff. For the most part, the DNA is what we call naked. But there are some chromatin proteins called SMC proteins that help compact. As you can see, there is compaction in this chromosome. When you splat it down on a slide, it starts to expand. And these SMC proteins help compact the DNA. And in prokaryotes, you get about a 4-fold compaction. I'm just showing you the structure of this protein because we talked about protein structure and function. And guess what? Here's one of those structural motifs, coil, that we talked about. What do coils do? What's coil for? It's protein-protein interaction. So Would you expect that this protein looks like this in its final folded form? Anybody? If I showed you this protein, what might you expect it to look like? Yeah, it's going to bind. It's going to bind to some kind of dimer, and these things do, and they're not sure exactly what that structure looks like. There's two ways to do it. This protein could dimerize with itself, so the two coils on the same protein could wrap around and make a structure like this, and then those dimers could interact at this hinge region. The other way it could do it is those two extended coils, both of those coils from one protein could wrap around the two coils from another protein and give you a structure that looks like this. The final structure looks like kind of this hinge with these two heads on it, but they don't know exactly how it's made. All right, coils here are making very, very strong protein-protein interaction. Yes? Yeah, so these head regions are sticking to the DNA, and there's a number of different ways that people draw this, but they stick to the DNA and pull those things together in a pack. Not very well understood. There are proteins very similar to this, so cohesins look pretty similar to this protein. Those are the proteins that are holding together the two replicated sisters. Yeah. Prokaryotes. We see very similar proteins in eukaryotes. All right. So in eukaryotes, there's a huge variety of proteins that are involved in binding to DNA. The DNA in eukaryotes is definitely not naked. It's coated with proteins. We're going to talk about one specific class today, and that's the histones. The primary component of chromatin is the histone. The histone forms into a structure called the nucleosome. This is all vocabulary that you guys need to know. The nucleosome is 8 histones bound together, making an octamer. All right, so it's a pretty complex structure. It looks kind of like a barrel, and I'll show it to you in a second. 8 different histones bound together in an octamer. They have a lot of this is the protein or the amino acid composition. They tend to have a lot of very basic amino acids in histones. Why? Who can tell me why you have a bunch of basic amino acids in histones? Yeah. DNA is an acid, right? So we have positively charged amino acid side chains on a protein complex that's going to bind to DNA, which has all the negative charges in the sugar phosphate backbone. All right, so what do these look like? So what we want you guys to remember is that histones are made up of an octamer. We're not going to ask you specific structural questions about the histone, but I want to show you how this thing fits together. There are actually four separate histones that make up this octamer. Don't ask me why. H2A, H2B, H3 and H4. H2 and H2A and H2B form a dimer. H3 and H4 also form a dimer. And then those dimers are hooked together into an octamer. There is an H1 in human cells, not in budding yeast, and it's not involved in this nucleosome structure. All right, so they come together, they form this octamer. 2 dimers of H2A, H2B, 2 dimers of H3, H4 to make the octamer. And then the DNA wraps around that structure. So this final structure, the histone octamer, is a nucleosome. Definition. All right.
Do you want this?
So not so long ago, Karen Luger, who's now at Colorado State. Yeah.
That's what he's doing.
Maybe can you do that?
It's a Facebook group. I'm gonna upload them all here.
I'm sorry?
Yeah.
Octamer. So this is how they assemble. I'm just showing you how they assemble. You get
Just
a
LEA.
dimer binds to another dimer, and
That's
then
not
you get
working.
8
Alright,
in that octamer. Right? Yeah.
Sorry.
That
What? What are you doing?
Yeah, so the question is that octamer form without the DNA being
Add me
there?
and then I'll add you to the group.
So there are enzymes that actually put nucleosomes onto DNA, and I can't tell you whether they're fully assembled before they add or not. They're actually, yeah, there are various structures for nucleosomes, and that can change it. All right, so the crystal structure was solved by not so long ago, 1997. And here's the histone octamer, the nucleosome, and we're looking at an edge on, or face on, and then when you look on an edge on, what you can see is the DNA will wrap about 1.5 times around that nucleosome. And then that's one way that you get compaction, is this wrapping. All right, so this is what I'm going to refer to as the first level of compaction. So we're talking about histones and their ability, the role here, the function of the histones we're going to start with is that they help compact the DNA. You need to compact it in order to stuff it into the nucleus. So a very long time ago, people recognized if you isolated DNA under some not very harsh conditions, meaning that you left a lot of the proteins stuck to it, you got this structure that people call beads on a string. These are all nucleosomes. So this is a DNA strand wrapped around a nucleosome, wrapped around another nucleosome, wrapped around another nucleosome. First level of compaction is this wrapping around the nucleosome. Here's what it looks like in cartoon form. So when you look at these beads on a string, there's the amount wrapped around the nucleosome. It tends to be about, depending on how much, depending on the conditions you isolate it in, about 170 base pairs per nucleosome. And then you have these linker sequences between the nucleosomes. All right, so this is the picture to remember. All right, so there's a linker between the nucleosomes, and the nucleosomes look like beads on a string. All right, second level of compaction gives you what they call the 30 nanometer fiber. So you take these beads on a string, and you stuff them and stuff them into another level of packing. You take all those beads on a string and you bring them together. And this forms a fiber called the 30 nanometer fiber. And you can actually see this with electron microscopy. These are all the nucleosomes right here that have now been packed together. Yes? There's different levels of packing in different organisms. So I'm gonna show you that we're gonna get to the very end. I'm gonna show you all the levels together. That's what I want to know. Okay? Did you have a question?
It's the second
The second level is 30 nanometer fiber.
No. So the question is the DNA? Is there anything specific about the DNA in the linker region? Yes and no. I mean, it is when it's extended, but not when it's all packed here. And remember, these linker regions aren't big, right? So genes are in the kbs. We're talking about hundreds of base pairs here. And we'll talk all about what happens to this nucleosome structure when you start transcribing or replicating DNA. All right. So now, in another layer of compaction, you take this 30 nanometer fiber and you stick it to a set of proteins called chromosome scaffolds. So proteinaceous scaffold's the best word for it, right? It gives structure to these chromosomes, and you start looping these chromosomes. And when you look at genes that are highly expressed, they're normally found out at the end of these chromosome loops. You have a chromosome loop attached to it. You know, you have this 30 nanometer fiber, and you're attached to that scaffold. It's going to be tough to get machinery in there to transcribe it. So often you bury genes that aren't being transcribed, or sequences that don't contain genes in these scaffold bindings. And here's a electron micrograph where they've actually detached the DNA from this chromosome scaffold. So these are a whole bunch of loops of DNA, and here's the scaffold. All right, so this is the money slide right here. All right, takes it all, puts it together. You start with a region of double-stranded DNA. First level compaction, beads on a string. Next level, those things pack in a 30 nanometer chromatin fiber. Okay? Then you take those fibers and you fold them up and put them on top of a scaffold. And then in mitosis, and only in mitosis, you have another level of compaction. So those cells, as we talked about, take their chromosomes and make them really, really small during mitosis. Everything just gets packed up. And we're not going to talk about that level today. But it's actually very, very significant amount of compaction.
Clearly you don't want to talk about it.
10,000 fold shorter.
Clearly you don't want to
than
talk
its
about it,
extended
so just Google it.
10,000 fold shorter in mitosis.
Monica Fox.
Questions? Okay, good. So, I'm going to admit, when I learned this in biology, I was bored to
Wait,
tears.
that's you?
There are people working on histones, and I'm thinking, okay, great. Who cares? Just about the most boring thing on the planet for me. Histones actually do something, and people discovered this relatively recently, that goes beyond compaction. They actually play a very, very important role in the regulation of gene expression. And that's when I started getting excited about histones. So, histones isn't just a barrel. It actually has these tails that hang off of each of these protein subunits. Okay, so these long, extended, random coil looking tails. And those tails hang out outside of the DNA. So this is the histone octamer. This is the DNA wrapped around it. They have these tails. So how might these tails play a role in controlling gene expression? They're highly basic, I'll tell you that. I'm going to regulate something. What am I going to want to do with those tails? Think they're doing anything, just hanging off, waving in the breeze? How would I make them do something? Yeah.
Put proteins on
I'd
them.
modify them. I'd stick them to other things. Change their structure. That's exactly what happens. Histone tails, and people have started calling this the histone code. Histone tails get modified out the wazoo. There's a whole bunch of enzymes that are sticking small molecules and other proteins and all kinds of stuff on histones. They get phosphorylated. You guys already talked about kinases. They get ubiquitinated. That's a small chain of proteins. Another enzyme adds that. They get acetylated. They get methylated. They get modified like crazy. The modifications are very, very specific. And they happen at very specific residues on these histone tails. That's the histone code. So when and where these modifications and what these modifications are in histone tails is recognized by a whole bunch of other proteins. So how can modification of histone tails affect transcription? Well, modification triggers conformational change. That conformational change can actually alter the packing of the DNA in these histones. So if you have very basic tails, basic meaning acid-base basic, they're probably interacting tightly with the sugar phosphate backbone, right? If I modify them, I can change the local charge density and make them interact less tightly. So some of these modifications actually change the conformation such that those histones are not packed so tightly on DNA. You're going to need to get rid of the histones in order to get a polymerase to transcribe through that double-stranded DNA. So modification of histones, they call it chromatin remodeling, is the buzzword you'll hear. Modification of histones can lead to either open chromatin, what we call open, which means less compacted, or closed chromatin, which can inhibit transcription. So regulation of how these histones bind, how tightly they bind, by modification can affect the chromatin structure in a region and really affect how well you can transcribe genes in that region. It's not just about transcription factors binding promoter regions, which we're going to tell you it's all about that. It's not. This is another level of regulation. All right, so I want to make two more definitions for you. Open chromatin, euchromatin is what you'll see in textbooks. There are large regions of your genome that exist in either euchromatin or heterochromatin. Heterochromatin is closed chromatin. It's generally not transcribed. There are huge regions of your genome that exist pretty much all the time in various cell types as heterochromatin. Why? Why do you have big chunks of your genome that are all tied up in heterochromatin? Why would you do that if you're a cell? Yes? You want to protect it, or you don't want it to be transcribed. Why not? Why have all those genes if you're not going to turn them on? Genes for other purposes? There are actually genes, a whole bunch of genes are wrapped up in heterochromatin. Why? Yeah. Exactly. So what's the difference between a white blood cell in my body and a skin cell? They all have the same genome, right? White blood cells aren't always activated. We talked about this before. What's the difference? Every cell in my body has the same genome. What's the difference between a white blood cell and a skin cell? Yeah. They're expressing different proteins at one level, right? So how do they do that? Yeah, there's different genes turned on or off. Right? So if I want to be a skin cell during development, as I go from a stem cell and I start to differentiate as the word into a skin cell, I want to turn on all the skin-specific genes, right? What do I want to do with all my white blood cell genes? I want to turn them off. This is a really effective way of getting large blocks of genes turned on or off. It's one way that an organism with all the very complex cell types that you have in your body that are all very, very different, a neuron looks completely different from a white blood cell. One of the ways cells can do that is by turning on large banks of genes and turning off large banks of genes using chromatin packing. Questions about that? Yeah. It's a great question. Does the same chromosome always have the same euchromatin and heterochromatin in every cell? What's your expectation based on what I just told you? Yeah, the answer is no. You can actually see sequences move from heterochromatic to euchromatic during development, especially early development.
Sorry,
development happens
Sorry.
throughout the organism.
Sorry, we
over
weren't
large
head
period of
boy
time.
at our
So there's a
schools.
lot of developmental pathways. Early after fertilization of an egg, there's a lot of replication, and then you start to, these lineages are formed, right? So there's reasons that you have all these different cell types. Development is the process by which you get all these cell types. Yes? Why is it called heterochromatin? See, it's the not interesting part. Originally.
Make sure I'm just gonna take that one.
I don't know. Other questions? Okay, we'll see you on Friday.
Yes.